Hello everyone, welcome back to the cell signaling course. If you remember, in our last class we discussed different types of two component system: hybrid, canonical, mutant variant of receptor, response regulators and so on in plant kingdom. With that we finished one of the first type of receptor mediated signaling which we would cover in this course, and that was receptor histidine kinase. If I recollect, we discussed so many things until now which included what are signals, what are their natures, what are the regulation on these signals, what are different receptors, different function which they perform, different enzymatic activity which they have, or something as a receptor which does not have an activity inherently present in itself, but it associates with certain molecule. There are channel proteins which are serving as a receptors. What are the domain requirement of these receptors, and what are the effector molecules which bring about signaling to its place where it is happening. We'll now discuss the next type of signaling mediated by the receptor having a specific activity towards serine and threonine. What it clearly suggests that we'll have a receptor which will have the catalytic activity directed towards the serine-threonine, and this is an activity which is mediated by kinase. So it's a phosphorylation event which will be noticed over there. If you recollect our earlier discussions in the classes, these are the most abundant type of kinases which are present in the living world. All different life forms, they have these kinases and largely deal with them for multiple of their physiological processes. In human alone, there are roughly 300 genes which code for serine-threonine kinases. Remember, when we were discussing about kinases, we said that they could be soluble or membrane bound. So among these 300 genes, you will have serine-threonine kinases which are soluble as well as receptor bound. So if you talk about the plant world, Arabidopsis has 600 genes which code for receptor serine-threonine kinases. What it tells you is that the plant kingdom deals with serine-threonine kinase for bulk of their physiological responses. We saw examples of histidine kinase when we discussed the two component system in plants, but what you will notice that there is a huge abundance of serine-threonine kinase in plant kingdom. Not only that, they have 600 genes which code for receptor and soluble serine-threonine kinases. There are genes which actually code for multiple of the signals. And what are the signals for these serine-threonine kinase? These are steroid hormones, these are small peptides which are there, and other active molecules. These serine-threonine kinases in plants, they are also called receptor-like kinases. The membrane-bound receptor which has the kinase activity towards serine and threonine is generally called RSTK, which is receptor serine-threonine kinase. In plant world, they are also known as receptor-like kinases, meaning kinase which was discovered to have a receptor property and also the kinase activity towards the serine and threonine residues. We will also see in the class to come that how this receptor-like kinases, they have a lot of variants. You remember in case of two component system, we saw variants where pseudo-kinases were there, pseudo-response regulators were there. We will see variations which exist in receptor-like kinases in the plant kingdom. As many as 1,000 genes are there which make peptides which are secreted as ligand for these serine-threonine kinase receptors. You can imagine that almost 1,600 genes are involved in coding for receptor serine-threonine kinase and receptor-like kinases, as well as the ligands which are peptidic in nature. Almost all aspects of the plant existence is mediated by signaling through receptor-like kinases, which involves disease resistance, cell growth, signaling, meristem activity, as well as something which is very critical for plants, which is called self-incompatibility. This is with respect to the pollination, whether the plant is going to show self-pollination or cross-pollination, that is being regulated by some of these kinases which are acting on the self-incompatibility process. If we see how these receptor-like kinases have come into existence, they have actually evolved from a set of proteins which are called receptor protein kinase. Or if I say, think about proteins which are receptor in nature and have kinase activity, from that bigger group of proteins the receptor-like kinases have come. We have discussed that this receptor kinase or the kinase activity can be directed towards multiple different types of molecules, biomolecules for that matter, whether it is amino acid, lipids, sugar, or nucleic acid. So, the kinases which have led to the RLKs are part of protein kinases because they will do the phosphorylation on amino acid residues. And within the amino acid residues, we saw that how preferably serine, threonine, tyrosine, and histidine residues were getting phosphorylated. So, the receptor-like kinases come under the class of serine-threonine kinases. Among them, you have these receptor-like kinases. So, these receptor-like kinases will be having catalytic activity of phosphorylating the serine or threonine residues. If you recollect, I always mentioned that the residue is fixed and sometimes the order in which they will be undergoing phosphorylation is also fixed. Now, there are further variations which exist in the receptor-like kinases, and that is where we were saying that variants of RLK. What we commonly understand that any of these receptor-like molecule will have domain organization, an extracellular domain, a transmembrane domain, and a cytosolic domain which has the kinase activity. So, in nutshell, you have minimal 3 domains on these receptor-like kinases. And now the variation comes from the fact that how these 3 domains are assembled in the final molecule. So, the one which meets all the requirement of a receptor with all these domain present is called TMRK or transmembrane receptor kinases. This is full receptor molecule which has extracellular domain, transmembrane domain, and the cytosolic domain. Then there are variants of RLKs which are called RLPs, receptor-like proteins. These receptor-like proteins, they have extracellular domain, they are embedded in the membrane, but they lack intracellular domain with kinase activity. Then you have a complementary of that which is called receptor-like cytoplasmic kinases. Here you will have these kinases with their catalytic cytoplasmic kinase activity domain and the transmembrane domain which is holding them into the membrane. They clearly lack extracellular domain, and these ones lack intracellular domain. So, when the signaling will be performed in the plants, you will see a combination of these molecules, a TMRK going with the RLP or TMRK going with RLCK. Together they will be ensuring that receptor is activated and signaling is perpetuating. Further, in this TMRK or transmembrane receptor kinases, there are multiple forms which have been identified. The ones which are actual kinase kind of property present over there have these two importantly conserved residues, arginine and aspartate. At a very precise location in their three-dimensional fold, they have arginine and aspartate residues. Those are called RD kinases. There are variants of this where arginine is absent, non-R, meaning arginine is absent, some other residue is present, and the aspartate residue is found over there. Such kinases are called non-RD kinases. Then there are examples of these TMRKs where both these conserved residues are missing, which basically makes them RD minus kinase. So you understood that from where, from a bigger class of protein kinases or receptor protein kinases, we have multiple of those examples leading to receptor like kinases, and then further they have the variations like RLPs and RLCKs, and then non-RD kinase or RD minus kinase. Now these receptor like kinases, I have already told you in the previous slide that they have this scenario. So you can simply understand that your TMRK will have an extracellular domain, a transmembrane domain and an intracellular domain. This is ligand binding and this is kinase domain. This is a scenario of a TMRK. If you talk about the RLP, you have a scenario like this where extracellular domain is present, transmembrane domain is present, but if you talk about this domain, it is absent. And when you will talk about RLCKs, you will have a situation where the kinase domain is present. Sometimes the soluble kinases which associate with the receptor, they are also referred as RLCKs. But classically, they should be tethered to the membrane to be called RLCK. So we will see combination of all these forms of the receptor like kinases coming into the fore for signaling. Now the diversity in the receptor comes from the fact that extracellular domain which is present, how is that arranged? I told you earlier that the receptor must have at least 3 domains. One, which is the intracellular domain, has a kinase activity, and that too is specific for serine and threonine residues. A transmembrane domain which is holding the entire receptor in the plasma membrane. Often they are single pass transmembrane entities, and then extracellular ligand binding domain which is the most diverse in nature. So what happens that when you have a receptor, their intracellular domain and transmembrane domain may look very similar, but the extracellular domain tells that how they will be appearing. What are different types of extracellular domain? If you recollect in receptor histidine kinases, in case of bacteria, we saw receptor with extracellular domain like JAF, which were responding to the cyclic di-GMP. Then we had PAS kind of extracellular domain which was responding to gaseous small molecule, and then we had another HAMP like domain. In case of plant, we saw that they were responding to cytokinin and ethylene. So basically the extracellular domain is driving the direction of the signaling. So let's see what are different types of extracellular domain which are present in receptor like kinases, which will help in generating a myriad response in plants. One of the first classified domain is called S domain, and this S comes from the self incompatibility. So in receptors which are in this class, they have an extracellular domain like this. If you can consider the extracellular domain. Remember, when we are talking about these receptors, we are talking about the complete receptor, which has an extracellular domain, and they are rich in the cysteine moiety. The other class is called LRR domain, where you have leucine repeat rich, where extracellular domain, if you consider as completely made up of repeats, you can say units like this will be there. That will help in the interaction, identification of the ligand and interaction with the other receptor for activation. Then there are domains which are called EGF-like domain. They have conserved cysteine residue and help in the pathogen response. EGF is called epidermal growth factor. When we will come to lot of signaling mediated by receptor tyrosine kinase in animal kingdom, we will see the presence of such a domain or such a molecule which is involved in the growth of epidermal tissues. So, structurally this extracellular domain of a receptor like kinase is similar to the epidermal growth factor receptor which is present in the animal world. You also have a domain which is called TNFR like domain. Again, this comes from the animal world, tumor necrosis factor receptor like domain, and this is involved in the epidermal differentiation. There is a lectin like domain which is present in plants, particularly because plants have lectin. They see different kind of signals which are rich in lectin. They actually perceive the oligosaccharide. In animal kingdom, the equivalent will be oligosaccharide recognizing or sugar recognizing receptors which are present over there, and we will see how those things are changing significantly in animal kingdom when we compare it to the plant kingdom. There is additional class of the extracellular domain which is called PR5 domain, which is basically pathogen response. So, this is important for pathogenic response or response to pathogens which are invading the bacteria or trying to gain an entry into the organism or the plants. So, simply if we think about the receptor like kinase signaling, what they very prominently do is they activate the MAP kinase pathway. This term MAP comes from mitogen activated protein. Again, this is a molecule which was very well studied in animals, where mitogen is a entity, small molecule, any protein, anything which has the capability of inducing cell division, cell growth. Basically, they are inducing mitotic activity inside the recipient cell or signaling cell. So, you would notice that we have been going back to animal kingdom to explain some of these domains and so on. The reason very simple is that they were first studied and classified in the animal kingdom, and later equivalent of those have been found in the plant kingdom. So, how this signaling works? You think about the receptor which is present on the cell surface. The two receptors come together and this dimerization event induces the phosphorylation. Remember, one of the first event is autophosphorylation of the receptor. This autophosphorylation of the receptor makes it active. Now, this activated receptor can directly phosphorylate an effector molecule, or it may activate another kinase in the system to activate the effector molecule. And the effector molecule here is nothing but the MAP kinase pathway. It is almost like a phosphor relay which you saw in two component system. hybrid two-component system. So the activation of receptor could be just because the two receptors come together, same type or different type. So you will see that in the receptor-like kinase signaling, you have homodimer as well as heterodimer forming and activating the receptor. So heterodimerization, homodimerization is common, as well as there is scenario where you will get this molecule in between. This is your receptor-like protein, which has the extracellular domain, transmembrane region, but lacks the cytoplasmic kinase activity. This helps in bringing the two molecules together, or this may not be required. It basically serves to activate or give proper response to the functional receptor, and thus it will culminate into the MAP kinase pathway. I mentioned about the phosphorylase. Let's see what exactly will be happening in the MAP kinase pathway. I told you MAP is your mitogen activated protein kinase. There are 3 of such proteins which are involved in this pathway or the cascade, if I say. Each one of them are serine-threonine kinase. Once they are activated by active receptor, the first kinase in the series is called MAP-KKK or MAP3K. This will be the first molecule which is activated. This will pass the signal to the next molecule, which is MAP2K, and finally you will have the last molecule, which is MAPK. This MAPK can phosphorylate the next molecule, which is final effector, or this may itself go inside the nucleus, and what it will do inside the nucleus? Activate the transcription. So you see the analogy of phosphorylase which I was telling. The phosphate which is on this molecule is going on this molecule. It's not the phosphate transfer, rather ATP will be utilized here, and the next kinase will be phosphorylated. The phosphorylation is making that particular kinase active. It looks for the substrate, utilizes ATP and phosphorylates the final molecule. In very early classes I told you this phosphorylation is a kind of switch to make the molecule active or inactive. In this particular scenario where we are discussing MAP kinase pathway, every one of these 3 kinases are getting activated by the presence of this phosphate group over there. So it is in some sense phosphorylase, but utilizing ATP at every step. Also in case of phosphorylase, which we saw in 2-component system, the phosphate was moving from histidine to aspartate, aspartate to histidine, and then finally to the aspartate. But here each of that phosphate residue is getting attached to the serine-threonine residue. Each of these phosphate is attaching to the serine or threonine amino acid. And this is how the signaling will be mediated in the plant system. So what we understood in the today's class is that plants are extensively dependent on receptor serine-threonine kinase signaling. The multiple physiological processes, you talk about root development, shoot development, plant growth, then something related to meristem, leaf development, and so on, multiple of them, even stomata development. That all is mediated by these molecules which are in the class of receptor serine-threonine kinase and fondly called receptor-like kinase in the plants. There are regular receptor-like kinases and there are variants where we saw receptor-like proteins and receptor-like cytoplasmic kinases. Further, homodimerization or heterodimerization is what expected from these receptors, and particularly when they heterodimerize in right combination, they will activate signaling which is desired by that final target cell. Thank you.